Yeah, thank you so much for the kind introduction. Yeah, I've been spending the last 10 years at Illumina developing deep learning tools for interpreting the non-coding genome. So today I'll be presenting a tool called PromoterAI, which we released last year to predict the functional impact of promoter mutations. Specifically, given a promoter mutation, does it increase gene expression, decrease gene expression, or doesn't have an impact? I'll start by giving an overview of the PromoterAI model architecture and training process. The input to the model is a 16 kb nucleotide sequence around the promoters. You can go higher, but because the focus of this project is mainly promoters and we don't really look into UTRs or enhancers, all of our results saturate at this particular length. So we kind of keep it at 16 kb. The output of the model, it shares a lot of similarities with other models in this category. So we predict for gene expression, we use CAGE, which is essentially gene expression at a nucleotide level resolution. And in addition to CAGE, we also predict histone modifications, DNAs, and TF binding at a nucleotide resolution around the transcription start sites. Over time, we have noticed internally, and we're also seeing external work saying that yeah, histones and TF binding, you probably don't need it anymore. But CAGE and accessibility are super important. Like, I mean, CAGE, you can either use CAGE or RNA-seq, but for DNAs, you can either use DNA-seq or ATAC-seq, but those two are very important, and the other two are negotiable. And in terms of the architecture of the model, we use a fully convolutional model. So this one, the architecture inspiration is a paper from a few years ago called Metaformer, where they argue that the reason why these transformer models work is not as much the attention operation, but the way the architecture is set up, where you have a token mixer with a residual connection followed by an FFN layer with a residual connection. They argue that you can replace the attention operation by any operation that mixes features across positions. And we tried several options ourselves, and in the end, including attention, and in the end, found depth-wise convolutions to be the most effective in terms of performance. And so we do get large coverage using dilations on top of the depth-wise convolutions. The training process as such, we train it in two phases. The very first phase is very similar to the other models in this category, where you do a simple sequence to histone, DNase, TF binding, and CAGE. But the main difference is every layer is a nucleotide level resolution, and we can afford it because the input lengths are only 20 kb. And the main contribution of this work is the second step, which is a fine-tuning process that achieves generalization across unseen genes and data sets and provides significant performance boost. So to do this, we first created a curated list of promoter variants with known large expression effects, meaning like they increase expression dramatically or decrease expression dramatically. And we use that to fine tune the model that came out of the first stage of the training process. And the way we do this fine tuning is we create two identical copies of the model. One copy gets the sequence with the reference allele, and the other copy gets the sequence with the alternate allele. So the output of these two copies will be, one will be the predictions for the ref allele, another will be the predictions for the alt allele. We take the average across all of these tracks to get a single score and fine tune the weight so that this score matches the known expression consequence. There are many ways to do fine tuning, but we have found this one to be the most effective in terms of how much performance improvement we get and also how robust it is to generalize across unseen genes and data sets. Our intuition here is that when you have when you provide like 20,000 nucleotides or more, there is so many features that correlate with the output that the model cannot really differentiate between correlating factors and causal factors, and it just picks up, it could pick up anything. But when you have a data set like this, where between the two branches, there's only one nucleotide that is different, but the output is completely different because these are like large expression altering variants. That means the model is now forced to attribute the change to exactly what is happening around the variant. So you are forced to focus more on the causal effects than the correlating factors. And to create this curated list of promoter variants with known expression consequence, we make use of GTEx data, which has both whole genome sequencing and multi-tissue RNA sequencing. And this part of the work project was handled by Nicole, who had done similar work for the GTEx project. So she re-implemented the pipeline that they used for the GTEx project, where you do a whole bunch of corrections for batch effects like expression PCs, genotype PCs, peer factors, and all of those. And when you like run the pipeline, setting the parameters so that you get so what we're really looking for here is, let's say, a singleton promoter variant, where like, okay, only one individual has that promoter variant, and that individual has an extremely aberrant gene expression for that gene across multiple tissues. And we try and see how many of such variants can we actually find. And when you run the pipeline, where you set the parameters of the pipeline so that by chance you will end up finding 1,000 such events, you will actually see that the pipeline will gives you about 2,500 variants in total. And the background, you can find it by like permutation analysis, where you can randomly assign genotypes to different individuals and see how many events can be caught. And when you essentially what's happening is when you randomly assign genomes to individuals and run this pipeline, you get 1,000 such events. And when you correctly assign the genotypes, you get 2,500 such events. So that kind of tells you like 1,500 events are real and the remaining 1,000 are kind of noise. The success of fine tuning really depends on the size and quality of the fine tuning data set. So we wanted something much cleaner. Um, so what we realized here was that when you really look at the actual gene expression, there are many factors that are contributing to the gene expression. One of them is promoters, then it can be cis-QTLs, and the other things are like trans factors. Something might be happening in some other gene that might be contributing to the gene expression of the current gene. And any variation that's attributable to cis-QTLs or the trans factors, they kind of muddle the signal that comes out of promoters. And so you get a cleaner signal if you can regress these things out. So we built, as a second step, like we built a model where you take all the cis-QTLs about 100 kb around the promoter and try to predict the gene expression using cis-QTLs. Whatever you can predict from the cis-QTLs, we regress it out because that has nothing to do with the rare promoter variation. So once you regress for those factors, you see that now when you run the pipeline with where you see 1,000 variants as background, you see about 3,000 variants for the correct when the right individual gets the right genotype. So it's become much cleaner. And finally, we also model trans effects by building a model where you try to predict the gene expression of each gene using gene expression of all other genes in other chromosomes. And so whatever you can model using this one, again, this has nothing to do with the promoter variation. So you can again regress these things out. And here's where we get the maximum boost. And once you regress all of these things out, you get 4,000 outlier variants where only thousands possible by chance. And this is all again still singletons, but you can extend it to rare variants. You can't go to common because of things like linkage disequilibrium, but you can do up to like allele count 8, and so you get you can kind of double the number of outlier variants as well. So that's what we use to train the final model. And in terms of evaluating these models, yeah, the variant interpretation is not yet a field where there are completely standard benchmarks that everyone uses. So I think as part of these projects, we also spend a lot of time creating benchmarks. So here for this project, we have 6 benchmarks spanning MPRA, EQTLs, proteomics, and RNA sequencing. Some of them are readily available, which we just had to download, and some of them we had to create completely from scratch, and some of them like we had to do a little bit of processing and so on. So and each benchmark has its own advantages and disadvantages. So when you cannot rely on a single benchmark to make decisions, but when you put all of them together, you kind of cover all of the bases. For each of these benchmarks, for example, you start with the KG5 saturation mutagenesis. So we try to solve do a 3-way classification. Given a variant, we ask whether the variant increases gene expression, decreases gene expression, or neither. Because standard metrics like ROC or precision recall are all for 2-way classification, what we do here is convert the 3-way classification into 3 2-way classification problems. Meaning, so we plot the ROC for an underexpression versus overexpression classification, or an underexpression versus null classification, or overexpression versus null classification. So the KG5 saturation benchmark, it's the most commonly used because the data is readily available. You can just download and you're ready to go. So in this case, we have 10 promoters for which there is saturation mutagenesis done. It's a very good data set for intra-gene effect prediction because for a given single gene, you have a lot of variants, but the downside is you have only 10 genes. So if your model doesn't do well on one of those genes, suddenly the performance can not look so good. And two others are MPRA benchmarks from Siraj et al., where they have a lot more promoters, like about 50 of them, where these promoters harbor either EQTL variants or non-GWAS hits. For all of them, again, for each of these variants, the score already comes from MPRA, and we try to we discretize the score based into like an under, over, or a null, and ask the model to predict them. The bottom row is where we had to do more work. So for the GTEx EQTL, we look at the GTEx like fine map DQTLs, like, and we require that the variants are fall in the promoter regions and are fine mapped in at least a couple of tissues. And the nulls are still associated variants, but fail fine mapping. But also more importantly, they belong to the same genes as the ones that fine map, so that like you don't reward the model for easy wins, like saying that some genes don't harbor EQTLs or some genes do. So this is all the hardest problems, where it's the same list of genes that have both the under, over, and null variants. For the UK Biobank proteomics and gel RNA was essentially like large multi-month projects, like where we had to start from scratch and create the entire benchmark ourselves. So UK Biobank proteomics, there's about 50,000 individuals for whom proteomics is done. So it's data set that's very high in terms of the number of individuals. But the downside there's only about 1,200 proteins for which the data is most reliable. So you get data for only those genes. So essentially, given that if a variant changes gene expression, then as a downstream effect, it is also expected to change the protein expression. So we also put these models through this task and see if they can predict the protein changes well or not. And gel RNA sequencing is a little bit different from GTEx and sensor. It's only blood, but you have 5,000 individuals, so it's much higher on the individual axis than the GTEx. So it's like you also have an RNA sequencing like target, whether your model's predictions can be validated using RNA sequencing or not. The more importantly, again, like I think when it comes to evaluating PromoterAI here, the fine tuning is a once done, and like the model is frozen. It's done on GTEx dataset during training, and then it's completely frozen. And all of these datasets were never seen during any part of the training process, including fine tuning. And all of these genes are also like unseen genes, so that like I think it's a perfect generalization for PromoterAI. A few different observations that we have seen. So one of them, Puffin, for example, has much weaker performance compared to other models. The only difference between Puffin and the other models, the main one, is they only model expression quantities and nothing else. Like Puffin, for example, its output tracks are just different forms of expression, like cage, rampage, and fair seek, and so on. So that's something that we have internally seen as well. You absolutely need to have ATAC seek in your output, and if you don't have that, you tend to lose performance. So histones and TFs are negotiable, but we suspect that I think the next generation of models will be sequenced to ATAC and something about expression, but you cannot just do only expression. And another interesting that we've seen is when you look at Chrome BPNet, for example, it's something that models only ATAC seek. And it's actually very, very competitive in all of the MPRA benchmarks, but it fails badly on actual like RNA-seq or protein sequencing benchmarks. So that's again another reason why you cannot just do only RNA-seq or only ATAC sequencing. So if you want a best performance across all of the different options, you seem to need both of them. And there's also many models coming from the David Kelly, like you see that each successive model is better than the previous one. And we are comfortably ahead of Borzoi, but also more recently, there is Alpha Genome that came out, and because we are from industry, we cannot evaluate it ourselves, but we are told that Alpha Genome falls somewhere between PromoterAI and Borzoi. It's worse than PromoterAI, but it closes the gap by, it's much closer to PromoterAI than it is to Borzoi, is what we are told by people who have run the benchmarks. And in the next few slides, like I will show you like a few different analysis we did to get a better understanding of what's happening in different parts of the training process. So I'll start with the gene expression outliers that we have created, and we try to get some understanding of why do these gene expression outliers actually change expression. So we did a whole bunch of analysis on what kind of motifs these outliers overlap. So we took all of these gene expression outliers, like, and just annotated all of the motifs they overlap and compared it with random promoter variants and see which are the motifs that are significantly enriched in outliers compared to background. And we do notice the usual suspects like tend to pop up, like, which is very, very reassuring, like YY1, ETS, you have the EBOX, E2F, TFAP, and all of those ones. And one thing that we noticed was all of the ones that were popping up in the underexpression often had an activating effect annotated, and all of the ones that popped up in overexpression tend to have a repressive effect. So there is when a variant can increase expression by either strengthening an activating motif or weakening a repressive motif. So we wanted to see if there's a preference for one or not. So what we did was we took all of the underexpression outliers, looked at the motifs they overlap, and tried to annotate them with the geo terms to see and stratify them on whether they overlap an activating motif or whether they overlap a repressing motif. What we did notice was when it comes to the underexpression outlier, so the pink one, so the quantity that I'm plotting here is the change in the motif strength between the ref and the alt alleles. So a positive value here means the variant strengthens the motif, and a negative value here means the variant weakens the motif. You do see that most of the underexpression outliers, the way it is being caused is it takes an activator motif, it overlaps an activator motif and weakens it. When you look at the overexpression outliers, the most common way was it overlaps a repressive motif and then it weakens it. But you also see quite a bit of signal for it overlapping an activating motif and then it strengthening it. But I think the main summary here is, yeah, the primary mode still seems to be weaken motifs. If it weakens an activating motif, it becomes an underexpression outlier. And if it weakens a repressive motif, it becomes an overexpression outlier. In terms of the PromoterAI model itself, we tried to do some visualization analysis to see what kind of understanding of promoter biology can we get out of PromoterAI. So what we did was we took the sequence of the promoter sequence of all of the 20,000 protein coding genes and passed them through the PromoterAI model, and took features at all of the intermediate layers and did cluster analysis on them. On the UMAP, if you don't do any coloring, you will only see one big blob. But if you do k-means, like, you see that it clusters into three major clusters. And these three of them have very distinct properties. So when you look at the blue one, for example, it's very, very high on the usual promoter marks like H3K27 acetyl and H3K4ME3. And it's very weak on the repressive mark H3K27ME3. And when you look at those genes, they tend to be ubiquitously expressed. Like, when you plot the number of GTEx tissues where these genes have expression, it's pretty much expressed in almost all of the tissues. The mean expression is very, very high, and the across tissue variation is very, very low. So the first cluster is essentially genes that are almost always expressed very well in all of the tissues. The second and third ones tend to be more tissue variable. Like, you see that the histone mark, the promoter histone marks are like much lower. The repressive marks are much higher. And when you look at the number of tissues where they're expressed, it changes a lot from 0 to 50. The per tissue expression means is also very lower, and the tissue variation tends to be much, much higher. And when we try to get a more precise understanding of each one of them, and when you look at the different genes for which we actually, when I say outlier genes here, it's all of those genes for which we have detected an outlier variant. We find that they almost always fall within the P1 category, which essentially tells us that, yeah, for us to be able to detect outliers, those genes have to be expressed in a lot of tissues. Otherwise, it's very, very difficult for us to detect outliers. So that means that, yeah, I think so all of our outlier genes, like, always tend to fall under one of the clusters. But again, this one was the one where we were kind of sure what that cluster was doing. The second one, we were able to pinpoint it once we found. So we downloaded from another paper, like, list of all regions where there's bivalent chromatin. And when we looked at all of the promoters where they overlap bivalent chromatin, so this is about 4,000 or so promoters. They almost always fell within P2, the second cluster, which is now the cluster where these are promoters with bivalent chromatin. The third one was a little bit harder to pinpoint, but given that there was too much variation happening across tissues, we suspect there might be something going on with enhancers. So what we did over there was we randomly took about 50,000 enhancer sequences from the genome and passed them through Promoter AI and plotted them on the same UMAP. And we found that they almost always fell on the third cluster. So that tells us that the 6,000 or so genes that or promoters that fall in the third cluster have very, very enhancer-like characteristics. So these might very well be enhancers that are just doubling up as promoters as well, meaning that they might be just be transcribing the sequence that is downstream of them. So those are the three main categories of promoters that Promoter AI described. And lastly, we also did some analysis on the fine tuning, specifically why it gives us so much improvement compared to the model without fine tuning. And so one of the things that we were noticing, I'm only showing one anecdote here, but you find like many, many, many such examples where this is happening. So this is one particular promoter where we did ISM on plus minus 100 basis of the promoter. In the first row is the ISM of the model before fine tuning, and the second row is the ISM of the model after fine tuning. You see one thing. So before fine tuning, you see that the model claims that variants that weaken ETS1 actually tend to increase gene expression. This is completely wrong because ETS is a activating factor and strengthening it is supposed to increase gene expression. So the right direction for ETS1 has to be negative. So this essentially tells you that like this ISM plot tells you that the model detected that ETS1 is actually doing something but got its direction completely wrong. And this is consistent with a couple of papers recently that highlighted these multitask models like having issues with getting the direction of motifs right. You see that after fine tuning, for this particular promoter, the model did two things. It completely switched the direction of the ETS1 to the right direction, which now tells you that hitting that weakening that motif will reduce the gene expression. And it also detected the presence of one more YY1. So fine tuning does a combination of two things. Like one, it fixes the direction of effect prediction of some of these motifs where the model before the fine tuning did not get the direction right, and it also finds new motifs where the model failed to find it before fine tuning. And while I'm showing one example here, we have shown many more anecdotes in the paper, and also not just anecdotes. You can do a systematic analysis of getting the direction of effect right for those motifs for which you know the direction. So what I'm showing here is on the x-axis is the model before fine tuning, y-axis is after fine tuning. And for all of we stratify the variants based on the known direction of effect, based on the TFs they overlap. And you see that across many, many different. So this also tells you that it's not just one or two motifs that the model is fixing because of fine tuning, but the improvement is broadly across different categories of motifs where after fine tuning, the direction of effect is much more consistent with what is known with biology than before fine tuning.
You
And
sure?
even yeah. Mhm.
There's a question on fine tuning. Maybe it's sure to have
Yes.
everybody on stage. What you call fine tuning here is when you have then fine tuned the model on the outliers, and
Mhm.
before fine tuning was a model more like learn across genes, like what we do with Borzoi,
Correct.
etc. Is that what you mean? Okay.
Yes. Mhm. Yeah, and
Okay.
when we did fine tuning on the outliers, again we set aside some chromosomes for validation, and that's what I'm using to show the plot on the right hand side here. Yes. Mhm.
Just try to understand a little bit. When you say fine tuning, you specifically use outlier gene expression data for the fine tuning here.
Yes, that is correct. Yeah. Mhm.
Okay, thank you.
Yeah, and also we can wanted to see fine tuning only like works on within specific distances or or it goes like even for like further away from the TSS. So for all of this project, like I think we mainly when we say promoters, we mean TSS plus minus 500. We don't go beyond that. And we do notice that within that plus minus 500 fine tuning in general increases the performance across the entire distance range, and it's not localized to any specific distance windows from the TSS. And lastly, one other interesting thing we found was when you look at those variants that at the plot on the right hand side, we stratify the variants into three categories based on whether the whether the variant had the same score before and after fine tuning, or whether fine tuning increased the score, or whether the fine tuning decreased the score. Decrease the score meaning it's predicting a much higher under expression outlier effect than after fine tuning than before fine tuning. And we did notice that for those kinds of variants, those variants tend to have very high phylopy values, which is very reassuring, because again, we never used phylopy anywhere in the fine tuning training process. But when you can see that the those specific variants for which the model actually changes the score, they're actually very, very conserved. That means that it tells you it's changing the score for the right kind of variants. And because we never showed phylopy during the training process, it tells you that it's actually picking up real biology and like not some hacky ways to get performance improvements. And in the next few slides, I will walk through some of the analysis we did to um, like show like how to validate the model's performance using human population sequencing data, and also how you can how it can be used to better understand the human genome. So in this slide, this is one analysis that we often do using nomad to evaluate different models. So the key principle here is that variants that actually do anything typically tend to get filtered out through natural selection, and over time tend to become rare and get thrown out of the population. So when you look at the common allele frequency variants that are common in the population, most of the time they tend to do nothing. So you can use that to get a way to get a metric to evaluate how well any predictive model that you have is doing. So the way to do it is you construct a 2 by 2 contingency table where one column will be common variants and one column will be rare variants, and one row will be your estimate for whatever is considered pathogenic, and the other row will be your guess for whatever is considered benign, for which you can use synonymous mutations, for example. And for whatever you consider as pathogenic, any model you train, you can put it through this test. So in this case, we use different intervals of PromoterAI scores. And whatever odds ratio you get out of this contingency table, if you do 1 minus 1 divided by the odds ratio, you get a metric that is reflective of how much selective pressure is there on variants that belong to that category. So as a sanity check, if you look at all of the variants where PromoterAI score is 0, you see that there's pretty much no depletion going on, which is a good sanity check. And as the PromoterAI scores keep increasing, whether overexpression or underexpression, you see that the selection pressure keeps on increasing. And at around like 0.5 scores, a PromoterAI score of 0.5, the selection pressure is almost as much as you will see when you use missense variants. And the ones with the highest or lowest PromoterAI scores, like +1, most overexpression or most underexpression, the selection pressure is almost as much as you see for the nonsense mutations. And you can also uh, because in all of these cases, it only tells you whether expression changes or not, and does not really tell anything about whether a gene is pathogenic or not. You can split the analysis based on the PLI of the genes, like whether it is a low PLI gene or a high PLI gene. You are expected to see a much higher selection pressure on genes which are high PLI, because they are important, and especially on the underexpression side, because these are genes that cannot tolerate a loss of function variants. And you see that the dark brown one compared to the light brown one tend to have much higher selection pressure than the genes with low PLI. And also, like using EQTL data, like using GTEx EQTLs, like where these are variants that find mapped on at least 2 tissues and fall on the promoter regions. On the y-axis is the actual effect size reported by GTEx, and x-axis is what is the PromoterAI score for those variants. You see a very healthy correlation of 0.56 between the two. And again, here fine mapping, it may not be perfect. So for a lot of the GTEx EQTLs in Siraj et al., like we have all of the candidate EQTLs, like we have MPRA scores already available, and you see that the model correlates even better with the MPRA scores, where on the y-axis is the value measured by MPRA for what the variant's effect size is, and on the x-axis is the PromoterAI scores, and this time it's 0.63, which is higher than 0.56. So we suspect it's because you don't have to deal with things like LD over here. And the third analysis over here, we use the UK Biobank dataset. And so over here is the UK Biobank proteomics dataset that I described previously. On the x-axis is the PromoterAI scores, and y-axis is how much change in the protein levels you see for individual carrying that promoter variant. Yeah, you again see a very healthy correlation. This time it's a little bit lower because Yeah, it's protein levels, and like we are only modeling promoter effects here. Like if we were modeling 3' UTRs and the other things as well, this correlation would have been a little bit higher. And as you would have, I forgot to mention this in the benchmarks, when you look at the difference between PromoterAI and Borzoi, the gap was closest in the UK biobank proteomics. So we suspect that yeah, when it comes to proteomics data, for you to do well, yeah, you also need to be able to model stability and things like that. So you're better off predicting not just the promoters, but also the 3' UTRs, like using much larger context sizes. And UK Bank also has a lot of quantitative traits, but this one's a little bit harder because it's not a one gene. It's not a single gene phenomenon. There might be many genes that are contributing to a specific quantitative trait. Using PTVs, we shortlisted about 100 gene phenotype pairs where that gene had the highest contribution towards a particular phenotype. And we put all of those genes through PromoterAI as well and see what kind of information you can get out of PromoterAI. For about 6 of those, we saw strong correlation. So for example, there's a strong correlation association between the GPT gene and alanine aminotransferase. On the x-axis, you see individuals carrying that particular promoter variant in the GPT gene, the promoter scores of it, and the y-axis is the actual trait value. Similarly, for HDL cholesterol, there's a couple of genes on the x-axis. You see the promoter score of the promoter variant, and y-axis is the cholesterol levels. There's 4 other things like where you see good correlations. And lastly, we are also very interested in rare disease. It's one of the primary use cases we envision. So for this, we use the Genomics England data set where there is about 100,000 individuals for whom there's a lot of rare disease metadata available. Specifically, like what we use over here is for each individual, depending on the disease they have a list of candidate genes already like annotated for them. This one's called Panel Lab. And we what we do is we run a pipeline with by assigning the right disease for each individual. So we call it a successful match if a variant falls for each given individual. If they will have, let's say, 50 or 100 genes that are candidate genes that explains the disease that they have. And if we end up finding a PromoterAI variant greater than a specific score in that gene, we end up calling it, okay, a successful diagnosis. And to estimate background, what we do is we shuffle the labels for different individuals. So you end up assigning somebody else's disease for each individual, and then we run this pipeline again to get the expected number of like background hits. So the expected divide by observed will give you an odds ratio, and the expected minus observed will give you the excess variant, which is the real number of variants being caught by the pipeline. So when you do run it through PromoterAI, you see that like when you assign the right phenotypes for the right individuals, you get 140 diagnoses. And if you apply, if you assign random phenotypes for each individuals, like you get 90 of them. So you get about 50 excess of them at a healthy 1.5 odds ratio. And this also gives us a way to evaluate the burden from different parts of the genome. So you can do this test using PTVs. You can do this test using different AI methods, like even from Illumina itself, like be it Splice AI, Primate AI, or Promoter AI. We do notice that if you use all of the different AI methods for interpretation, you can pretty much double the throughput you get from using only PTVs. So that's the real value, like when you get all of these different AI methods. And the non-coding itself, like when you take Splice AI plus PromoterAI, it's about 20% in total. And PromoterAI itself, like, is about 6% is kind of what is the genetic burden of these promoter variants on rare disease. And lastly, we also looked at ClinVar variants. Like over here, ClinVar is mostly coding variants. Like I think for promoters, we found only about 50 variants in the promoter regions that are like pathogenic and like actually non-coding. And for benign, like we find a similar number of variants, and we also used Nomad Common to like further boost the benign variants in those only in those genes. So And again, like you can get an AUC of like benign versus common. And we've shown several examples in the papers. I'm highlighting a couple of them here. One of them is in the promoter of the APC gene. You see like there's a variant that hits the YY1 motif, and you see that when you do the ISM analysis, only inside the YY1 motif the scores are very, very high, and as soon as you get out of the motif, the scores become very low, which essentially tells you that okay, the model thinks that it's weakening the motif there, that is problematic. And also when you look at the phyllo P, you see that the phyllo P is very high only within that motif, and outside that like it kind of like goes away. And you can also look at the TF-ChIP-seq data there, and you see that YY1 tends to have a very strong binding peak over there. So there's different ways to get evidence that, like this is a very real prediction. And there's similarly many other motifs for which you see all three of them go through. And lastly, one other thing that I want to point out, yeah, there is again like the two papers that I mentioned before. Like I mean, do mention that these models tend to have a lot of issues with personal genome prediction. And there are some asterisks there. One of the things that I think like often gets mentioned is that these models are very good at intergene prediction, but not so much intragene predictions. We do not believe that is true. Like for example, when you take the KG5 promoter variants, and why I'm saying KG5 here is because that's the one that has the highest number of intragene variants. Like for each promoter, you see like there's close to 1,000 variants almost. So you can plot all metrics intragene. Like for example, I only take the TERT gene. There's 743 variants. I now have the PromoterAI scores for each of those 743 variants and correlate them with the MPRA scores. I can do this with Informer too, or I can do this with PromoterAI. And the PromoterAI scores are consistently better, but more important, it's not that none of these models work. They all have very healthy intragene correlations. But it is it is true that when you try to correlate across RNA-seq, you do get like zero correlation. So what is really happening? Like I think we found evidence in the following. So one of them is for all of the variants for which we know the ground truth, we try to stratify them based on the TSS distance and try to see how much correlation you get. One of the things that we see is that these models work incredibly well, like in a very short window around the promoters. The best performance is about like 200 bases on either side of the TSS, and you get a good performance up to plus minus 500 bases of the TSS, but like it crashes down all the way to zero. Like after you go 1,000 bases of the TSS, like after that, there's no predictive performance. So when you try to do personal genome prediction, if you use these models to annotate like 100 KB worth of variants, like most of the variants, the scores assigned by these models are not reliable. So as long as you stick to within the promoters, these models have very high utility. And another thing that we noticed was RNA-seq data is inherently very noisy. Like when we the what you see in blue color here is when you use variants for which they only occur once in the population. The one in orange is for variants that occur at least twice in the population. So now you get some sort of averaging going so that you can average the data across two RNA-seq samples. You see that suddenly the correlation almost doubles like within the strong regions, which also tells you that like if you're trying to do personal genome prediction with just a single RNA-seq sample, like the RNA-seq itself is very, very noisy. But if you try to do some sort of averaging, I mean, this one, like we've like you the thing saturates after about 5 samples, but the biggest improvement you get is from 1 to 2 samples where it severely denoises the RNA-seq and the correlations tend to go up dramatically, like within the plus minus 100 window. And yeah, I think to summarize the project, yeah, I think we have a model called PromoterAI, which predicts expression consequence of promoter variants. And so it's trained via 2 stages. The very first stage shares a lot of similarities with the other models in this category, and it's no better or worse than the other categories, like with the very first stage. It's kind of similar, but the main contribution is the second step of fine tuning using the outliers, which gives us a significant boost that also generalizes to unseen genes and data sets. There's many ways to do the fine tuning. The most effective way is to do it via the twin network setup that I mentioned before. And we have also shown performance of across benchmarks spanning RNA-seq QTLs, proteomics, and MPRA, and validated it on Nomad, UK Biobank, Genomics England, and ClinVar. The main takeaway is, I think we see a strong performance. Again, we don't speak about any tissue-specific prediction here. So all of these variants detected by PromoterAI are ones that broadly alter expression across multiple tissues. The main thing is, the strong performance is at promoters, and we are defining promoters as the TSS location plus minus 500. And we do not recommend using these models outside that window. Within that window, it's very, very reliable. Um, in terms of the data availability, I think all of the fine-tuning data and the benchmarks are all already publicly available. The a couple of data sets so we also have scores for every single, so there's 87,000 TSSs for protein coding genes, and for each one of them, we have scores for plus minus 500 bases every single variant. That one's also freely available for academic purposes. Just the only one thing is it's behind, like you have to sign an academic user agreement. It's a 30-second process. It's in the GitHub page. So like if you just go and if you need that data, like just go ahead and sign that agreement. I would immediately share the data set with you. And again, while I'm the one presenting here, there's many, so it's a multi-people, multi-year project. Have listed all of them here. And with that, I'd like to thank the inviters and happy to take any questions.